Hello, everyone. Welcome. And as Gregor has introduced about this lecture, so I will give 2 lectures on human motor systems today as next week. My lecture, so I will encourage you to interrupt me. So if you have any questions, you just unmute yourself and ask. Otherwise, we can also discuss the material after the lecture is done. So I designed my lecture concerning mainly 2 aspects of human motor system. So the first is the basic anatomy and neurophysiology of the motor system. So these are basic descriptions and textbook knowledge. So and the second part is about more like a mechanistic level. So it's more like a theory and modeling. So that this aspect is really requires something of thinking, and it's also related with exercise and exams. And regarding the first aspect about the basic anatomy and physiology, neurophysiology, I would like to recommend the textbook here, the first 3 textbook. So I highly recommend the first one, the bare textbook, Neuroscience Exploring the Brain. So this is the entrance level. So if you want to know some basic physiology about the motor system, you could read this book. It's very easy to read, and I've been really enjoying to read this book. And the second, by the Kandel, Principles of Neuroscience. And this is a more advanced textbook, so it contains more details and much deeper knowledge. And the third one is more complementary, so less important. So the many of the figures of my lecture are taken from the bare neuroscience textbook. And concerning the second aspect of motor system about the mechanistic level, so we are talking about motor control theories. And then I would let you refer to the 4th textbook by Latache, Fundamentals of Motor Control. And this book covers the state-of-the-art theories about motor control. So if you are interested in theory and modeling, you could look at this book. It's also very easy to read and a very interesting book. Okay, so I think let's start the slides. We have been learning robotic control in the previous lectures. So at the very beginning, I would like to show you a video about robot control. This is one of the most advanced robots currently from the German Aerospace Center. We see that it can do the manipulation task. It opens a bottle and put the material in the cup. It can move the joints of arm and fingers. And later it will also pour water to make a drink. So this is quite impressive work. And but still we notice that the robot is actually not doing so flexible movement like the humans do. Particularly, it's very slow. Actually, this movie I have already adjusted twice the speed for display, and because I want to make the movie shorter. So in reality, the robot moves much slower than this. And one big problem about robot control is it cannot be generalized, which means if you have another task, if you let this robot doing a different movement task, you have to design everything new. So you have to reprogram, and this is a completely new research program. And now let's look at humans. So I will show you a video about a young kid who is participating in the stacking cup competition, and we'll see how he moves. So this guy did this so complicated manipulation of the cups within 5 seconds. And so we see clearly our human beings are much better in movement production and movement control than robots. However, if we compare the robot and the human, concerning the motor elements, we see that actually human motor system is not optimally well equipped for motor control. First, let's consider the robot has, as you know, this very powerful torque motor. So you give the electrical current as the input to the motor, and then it gives out the desired force that you want to produce. And this transition is usually very fast. So the torque motor will respond immediately to its input. But in humans, we have the muscles. Muscles are soft. So if you give input. So what stimulates muscle is the neurons. We will see later. So if we give a neural input to the muscle generates force very slow. And that's why we see these sluggish muscles. And the question is how we control this sluggish muscle and soft muscles to produce so many fast and complex movement. And the second aspect is about conduction delay. And in robots, the conduction, the transformation of control signals is through the electrical wires, which have practically zero delay. So we can almost neglect this delay. But if we look at human nervous system, our conduction of signals are through neurons in our body. And the neural transfer is much far slower than electrical wire. And let's consider the most fast loop within our body when we control the skeletal muscle from our brain. The most fast loop takes at least 20 milliseconds. So if you take this 120 milliseconds delay for robot control, it never be tolerable. So you can never control a robot like this. But human still works much better than robot. And the third thing about this movement accuracy is about these sensors. In robots, we have these high-tech sensors. We have very accurate measurement. Basically, whatever we want to measure, we can measure position, velocity, force, and so on. And in humans, we have also have sensors, but these sensors are very noisy because we have noise everywhere from the environment, from our muscles, and from the sensory feedbacks. So how do we account for this noise? And in particular, we have multiple sources of sensory signals from different sensory sources. So how do we integrate these sensory signals to make an accurate estimate of our own movement? So concerning this aspect, the question is how humans produce movement. Unfortunately, I cannot give you a definite answer because the field is relatively young and there are a lot of unknowns in human motor control, but I will try to cover some basic knowledge and what we are know yet to tell you in this lecture. So we'll have, let's have an overview of the human motor system. There are mainly three parts of the system. So the brain, as you see here, and the spinal cord. And in the lower level are the muscles. Usually we call the brain and spinal cord together as the central nervous system, and short for CNS. So in this lecture, for today's lecture, I will start from the muscle level and I will talk about the spinal cord. And in the next lecture, we will go to the more complex brain. So let's start from the muscles. I want to introduce this very famous neurophysiologist, Sheridan. He's a Nobel Prize winner about 100 years ago. He set the neurological foundation for motor control. And currently, our current understanding of the neural basis of motor control is still largely based on the Sheridanian world. And as Sheridan already emphasized, the muscle is the only actuator for movement production. So let's look at muscles. So here is, we see that if we take one muscle, we see the muscle consists of many muscle fibers. And for each muscle fiber, we see there's one axon terminate on the surface of muscle fiber. And these axons are from the motor neurons from the CNS, central nervous system, so from the spinal cord or from the brain. And I assume that most of you know what a neuron is. As shown on the right, this is a typical structure of a neuron, although it's not always like this. There are some other types of neuron, but let's consider this as represent the structure of a motor neuron. So one neuron, the inputs to the neurons, receiving inputs is expanded dendrites. It's like the trees with branches. So it receives other input from the surrounding neurons. And the center of neuron is the cell body. So the transmission of the signals is through the axon. Usually, motor neuron has very long axons. And on these axons, they are wrapping around this material called a myelin sheet. The function of this myelin sheet is to accelerate the conduction speed along axons. But even though they have this acceleration, the neuron conduction of signals is still very low. So the most fast speed is about 50 seconds, 50 meters per second. So let's consider if we want to send a signal from our brain to our arm muscles, let's account for 1 meter and divide it by 50. That means it's about 20 milliseconds delay if you want to control a muscle. So, okay, so and then this is about the neurons, motor neurons. So let's see this alpha motor neuron is the motor neuron that directly innervate the muscle fibers. As we see before, each muscle fiber is innervated by the one axial terminal from alpha motor neuron, while one alpha motor neuron can innervate multiple muscle fibers. And this consists the minimum unit of movement production is called a motor unit. And on the right side, each muscle can be innervated by multiple alpha motor neurons, and these motor neurons formed the motor neuron pool. So this is the basic structure of muscle and motor neurons. So now let's look a little bit deeper to each individual muscle fibers. So here is one muscle fiber. So if we look at more in detail, so the surface, the muscle fiber has a membrane like the surface, and inside the muscle fiber, there are many even smaller muscle fibers called myofibers. So these columns, and around each myofibers, there's like a circle. It's called T-tubulates. So the function of T-tubulates is to transmit the innervation or the activation from outside into inside the muscle, the myofibers. So as you remember, the axon terminates on the surface. So once the axon is activated, it sends it generates an actual potential, so depolarize the membrane. So and this mechanism will induce a flow of the calcium, the L through the L channels. So the calcium will come into the muscle fibers, and then what happens next? So when the calcium comes in, it interacts with the inside with the myofibers. So here we still look a little bit more detail about the myofibers. So if we look we took a slide, so we see the myofiber consists other components. There's a thin filaments and thick filaments. The thin filaments is usually called actin, so it's here, and the thin elements is called myosin. So they structured like parallelly. So usually in the relaxed state, they are separate, but they are quite close to each other. So once the calcium comes in, the calcium will bind together with the protein, which is called troponin, and the troponin is attached on the topomyosin. So once this calcium is binding with troponin, it will induce a change of topomyosin's shape. So the topomyosin will rotate, will change its structure. And this change will lead to the binding with the head of myosin together with the tropomyosin. So, and then the head of myosin will bend. So as shown here. So for example, it was 90 degrees before, and then it will have a smaller angle, about 50 degrees. And this, for this one cycle is actually a very small movement of the muscle. It's just about 1 nanometer. But since the calcium is keep coming in, so it's a continuous, this circle continues. So that's why this head will continues moving to generate muscle contraction. And this is actually a very simplified picture. So if you want to know details, it is actually much more complicated than this figure shows in the molecular level. So if you're interested, you go to the textbook I referred to before. So, okay, so now let's come out of the micro level and let's go consider the muscle as a whole. Let's take a isolated muscle and assuming, of course, there are many motor neurons, but let's assume for simplicity, there's one motor neuron innervate this muscle. And well, let's see how the muscle generate force in the muscle level. So if this motor neuron will generate action potentials, I'm not sure if you know what is actual potentials. They are the electric pulses that conducted through the neural axons. But the actual potentials, the frequency of action potentials depending on how active the neuron is. And also only when the neural membrane potential exceeds a certain electrical threshold, it gives out actual potential. Otherwise, there's nothing, there's no output. So with lower frequency of action potentials, we see they generate small peaks of muscle forces. They are separate peaks. However, if we increase the frequency of actual potentials, we see these peaks summed up. This is called a summation. So when the frequency is sufficient high, we see the muscle force generation is becoming smooth. Okay. So next, we will look at the spinal cord because the muscle is directly connected with the human spinal cord. The human spinal cord is an extension from the brain. So here we see this, it can be divided into different sections. The cervical section usually controls arm muscles, or shoulder or arms. And the thoracic section controls the trunk muscles. The lumbar cord section controls the leg muscles, and the sacral cord section controls the hip muscles. So usually we are more interested in the arm and legs for the limb movement. And here I also want to explain that there is a nerve called spinal nerves. So this is not a nerve, but actually a bundle of nerves. So this spinal nerves is actually bidirectional. So they both transfer signal from spinal cord to the muscles and vice versa. And this consists of this bidirectional is called motor and sensory pathways. So so motor pathway means you go out from the spinal cord. Let me first explain the structure of the spinal cord. We see it in the middle of the spinal cord, there's a H-shaped structure. It is called gray matter. And it consists mainly of the cell bodies. There are also many interneurons inside, but we will explain that later. And outside the gray matter, it is called white matter. So these are mainly axons of the neuron. So for these neurons are usually from the brain. So the cell bodies of this neuron is located in the brain, and the brain's these neurons sending axons down along the spinal cord, which forms the white matter. And the motor pathway is these motor neurons are located in the ventral horn of the spinal cord gray matter. Ventral means in front, and the dorsal means back. So the ventral horn, we have alpha motor neurons, and it's sending axons through the spinal nerve and innervate motor fibers. And in the opposite way, there's also sensory signals from outside and going through the spinal nerve and come back to the spinal cord. So these sensory receptors can have different types. So you can have sensory receptors which is sensitive to muscle length, velocity, or force, or even temperature, touch, pain, and so on. So many types of receptors. And interestingly, these sensory cells are not located outside or inside the inside the spinal cord. It is located in a place where it's called dorsal root ganglion. So it's here inside the spinal nerve, the cell body. So this signal, this basically one axon is transferred directly from the outside to the spine into the spinal cord. So now let's talk about alpha motor neurons. This is very important. Alpha motor neuron directly innervate muscle fibers, and we'll look at what are the inputs to the alpha motor neurons. So we have talked about sensory input from muscle spindles. And there's also input from spinal interneurons. As I said, there's still a lot of unknowns about interneurons. So many research is going on about to study the function of interneurons. In this lecture, I will also later mention some type of interneurons that we know the function, but still there are a lot of things going on now. And there's the third source of input is the neuron from the upper level, so from the brain. And this kind of the descending control of the alpha motor neurons. Okay, so this is important to know these three sources of the input. And now let's look at another structure called muscle spindle. Muscle spindle is actually located within the muscle. You see this structure, the two ends of the muscle spindle is thin and in the middle of is more thick. So that's why it's called a spindle. So what is muscle spindle doing? So you see in the muscle spindle, they are wrapping a lot of neural axons. These are the sensory neurons. And we will see how this is from next page. So in the muscle spindles, these neurons are sensory neurons, and also the muscle spindle actually forms the intrafusal muscle fibers. So what we have talked before is called extrafusal muscle fibers. These extrafusal muscle fibers directly generate force, but the intrafusal muscle fibers do not generate force. It only serve as a sensory function. And there's another type of motor neuron called a gamma motor neuron. It's also located, the cell body also located in the ventral horn of the gray matter, like the alpha motor neuron. However, this gamma motor neuron innervate the intrafusal muscle fibers. And what this alpha motor neuron, gamma motor neuron doing? So let's see in the next slide. So this is about the function of gamma motor neuron. Let's take an example. So let's take a muscle, isolated muscle. So here we plot the extrafusal fibers and the intrafusal fibers separately for clarification. And when we attach the weight to the muscle is lengthened. And simultaneously, the intrafusal muscle fiber is also lengthened. And then it becomes tight. So the muscle spindle will give out a sensory signal. And this, by the way, this sensory axon is called 1A fiber, so or 1A axon. So it senses the change, the length change of the muscle fibers, and which is the same as the length change of the muscle. So in this case, the sensory signal gives out information reporting the muscle length. And in the next condition, if we here, this is an alpha motor neuron. If we let it stimulate the muscle, so the muscle contracts and becomes shorter. Because the muscle spindle is attached together with the length, so its length will change together with the muscle. However, the muscle spindle becomes shorter, and so these 1A axons becomes also loose, so they are not holding the muscle spindle. And in this case, it can no longer sense any length information, because it's not grasping the muscle spindle. But if in the next condition, if when we activate the alpha motor neuron, we simultaneously activate the gamma motor neuron, which innervate the muscle spindle. So the muscle spindle becomes tight again. And then these 1A axons again can sense the length of the muscle to feedback to the nervous system. So here we see if we adjust the gamma motor neuron activity, it will adjust the sensitivity of the muscle spindle. So it will adjust the sensitivity of muscle length sensory feedback. So this is about the gamma motor neuron function. This is basically a scheme that showing how this works, as I have explained. There's a phenomenon called alpha-gamma coactivation. And so once your brain sending a command down to the spinal cord, the activation command sends both to alpha motor neuron and the gamma motor neuron. Alpha motor neuron directly innervate extrafusal muscle fibers, and gamma motor neuron innervate muscle spindles. And this muscle spindle gives the sensory feedback to the alpha motor neuron. So this is actually a reflex loop. So we will talk about this later in more detail. And this is kind of a feedback loop. So on the right side, there's an experimental demonstration of gamma motor neuron functions. And what is done by this experiment is they do the finger flexion movement. As we see here, the kinematics, so they flex the finger. And the EMG is electromyograph, so this represents the muscle activation. And this is a flexor muscle of the finger. So once you flex, you do muscle flexion, and then you have activity in muscle flexor muscles. And this, the next two plots represent the sensory feedback. So you see when you do the move, you shorten the muscle, you still get the activity in the sensory signals. So what does it mean? Because if you assume that if you don't have gamma motor neuron or it doesn't do anything, when you shorten the muscle, the muscle spindle should not sense any information. However, here what we record is it shows that the muscle spindle did sense something. So that this provides evidence showing that the gamma motor neuron should be active at the same time to make the muscle spindle tight. Unfortunately, we cannot directly recording the gamma motor neuron activity because the gamma motor neuron is very small, so it's very difficult to record. And that's why they use this indirect evidence for this gamma motor neuron activation. Okay, so now let's come to the reflex section. This is a basic structure of the reflex. Actually, this scheme is discovered many years ago, and first demonstrated by Charles Sheridan, who won the Nobel Prize for the discovery of these mechanisms. But it's about 100 years ago, and so here we know that there's a muscle spindle which connected with one axon sensing the muscle length signals, and there are alpha motor neuron innervating the muscle fibers. Assuming that your muscle is stretched, so if passively stretched muscle, the muscle becomes longer, and then the one axon is active, and this axon will send excitatory input to the alpha motor neuron to innervate, to active this muscle. So the muscle becomes contract and becomes shorter. And so in this case, this mechanism resists the change of muscle length, right? And on the opposite side, so we have this so-called inhibitory interneuron. So once this muscle is stretched, this excitation goes through the inhibitor neurons to inhibit the opposite muscle. The opposite muscle also called antagonist, and this muscle is called agonist. And it also simultaneously inhibits the antagonist, and this makes sure that your muscle is not further stretched. So this kind of two mechanisms called stretch reflex and reciprocal inhibition. Okay, there are even more complex reflex loop. Let's look at this flexor withdrawal reflex. Assuming that you step on a substack, so here, and then you feel a pain. What you do immediately is you withdraw your feet. And usually this is done very fast, even before you're consciously aware of the pain. And this mechanism is done through the spinal loop, which have relatively shorter delays. So here, you have pain receptors in your toes. And once the pain receptors sense this information about pain, it will active these excitatory interneurons. And these interneurons will further excite the corresponding flexor muscles. So there are several flexor muscles to bend the knee joint. So in this way, the muscle becomes shorter and your feet will drop back. So this is the mechanism for reaction to a pain. But let's consider, so you have a pain on the left side, or maybe here, let's go, if you look at directly, this is the right side. Let's take this left side. So if you have a pain on the right side, but you have to do something with the other leg. If you don't do anything about this leg, you will fall. So what you do is you do flexion movement of this leg, and actually you will do an extension movement of the other leg. And how this can be done? So this will stabilize your posture. So here is basically what we have talked about. So this flexor withdrawal reflex, but the pain from the right leg simultaneously goes to the opposite side. So this pain receptor will active another inhibitory motor neurons, and as a consequence, the flexor will be relaxed, and the extensor will be excited, and this leads to a extension movement of the other leg to make sure you are standing, but not falling. There are also other mechanisms about reflex. This is the Golgi tendon organ. So this tendon is connecting the bone together with the muscles. And there are sensory receptors in the Golgi tendon organ. And this is called a 1B axon. It senses the force in the Golgi tendon organ and sends back to the spinal cord. And then through an inhibitory interneuron, it will inhibit the alpha motor neuron. So that means once your muscle become active, then you have force on the tendons, and this force will inhibit your muscle. So this Golgi tendon reflex always resist the force increase. And that is particularly useful when you exercise very large force. So it may damage your muscles. So, but if you have this mechanism, it makes sure that your force do not go to a higher limit to protect your muscles. Okay, and now we will have a more complicated reflex. So here we have talked about reciprocal inhibition before. So here is what we have known. So we have muscle spindle feedback to the back to the muscle, to the same muscle, and through an inhibitory interneuron to the opposite muscle. So this is what we have just learned about stretch reflex and reciprocal inhibition. But what is more here, we see these dashed pathways. These are signals from the brain, from the cortical spinal system, so the upper level of the spinal cord. And this means the brain can modulate this, so they terminate on these interneurons and also on the alpha motor neuron. So this means the brain, the descending command, can modulate the sensitivity of this feedback. So how much feedback I want. For example, if I don't want this feedback, I can just send an inhibition command to block the feedback. So this is a more sophisticated, more complex control, descending control of these inhibitory neurons. And there's another type of cell, it's called in the spinal cord, it's called the Ranson cell. The Ranson cell is an inhibitory. So here you have a muscle, and once this muscle, the alpha motor neuron innervate a muscle, it simultaneously also innervate the Ranson cell. The Ranson cell becomes active and send an inhibitory input to the same alpha motor neuron. So it's like kind of a recurrent loop. So it also decreases the activity of motor neurons. And simultaneously, it always also sends the inhibitory input to the interneuron that connects with the opposite muscle. And so they have a opposite effect on the opposite muscle. So, however, this Ranson cell is we still don't know what exactly the function of the Ranson cell is, but we only have this demonstration here. And I also want to mention that actually many of this reflex loop is done in the animal experiment. particularly in cats and rats, because in human, you are usually not allowed to put electrodes in the spinal cord. Okay, we have go through many types of reflex. This is a modeling study, and this study takes all types of reflex together, and they do a model of this. For example, they have this 1A axon, which takes the muscle spindle signal going to 1A, and muscle spindle signal come back to the alpha motor neuron, and alpha motor neuron through the 1A interneuron, it comes to the opposite muscle. So this is the stretch reflex and reciprocal inhibition. There are also 1B axon, which is the Golgi tendon reflex, and R is the rational cell. Actually, there's even more other kinds of neurons in the spinal cord. For example, this PN, I haven't talked about. This is the propriospinal neurons, which travels along the spinal cord. And it's also, the function of this neuron also largely unknown. And there's also another mechanism called presynaptic control. This is also kind of a descending control of the neural activity. So there are a lot of things going on there. But in this study, they took everything together and take a model of the spinal mechanisms. So this is a very complex loop. But the question is, what are the inputs to this complex neural network? So in this study, they assume that they have two types of input. One is a go signal, one is a set signal. The set command is define the baseline activity of this network. And to initiate a movement, they will send a go signal. And in this study, they did the go as a step function. So they just took the simplest form. And they show that it can produce some reasonable movement. But this is still, again, an assumption of the descending input. So we don't know what this descending input is. Actually, I will introduce later. Currently, in our lab, our job is to find what the descending input should be to this complex neural network. So I will talk about this later. Okay. So what still needs to be done? What is missing now? So I haven't talked about a lot about the human experiments. So experiments are important. So we always have to measure something from the human movement or recording some data, and we want to extract some features from this data to see what is the characteristic of the human movement. But on the other side, we can not just measure everything we want. So we have to understand what we measure and why we are measuring. And for that, we need to have a theory or have a model to guide our experiment. And for the theory of motor control, the first point, the most important point, is to define a control variable. So what is controlled? For example, in robots, you can say I control position, I control force, I control stiffness, and so on. But in human, what is controlled? So that is actually a very big question in the field. We will look at this later. And I want to quickly go through different types of human movements. So generally, it can be divided to involuntary movement and voluntary movement. So involuntary movement is usually the reflexive movement, and usually without consciousness aware of what you are doing. So you are not changing anything that you are producing. For example, if you lift a very heavy bag, plastic bag, and suddenly the plastic bag is broken, the bag drops down, and then because the weight is disappears, so you will your arm will lift up. So this is kind of the reflexive movement. And voluntary is more common in the daily life. So you do reaching movement and so on. And we can study different segments, different parts of the body. So we can study arm movement. So arm movement is mostly studied in the field of human motor control. And we can also study locomotion, the walking, and this is mainly concerning the leg muscles. And in 2 weeks, Rashid will give you a lecture especially about this locomotion. So he will talk a lot details on the leg muscle control. There are also eye muscles. So this may be less familiar to you because if you look at the eye, it actually also has a very fine structure of the muscles. So I will not go to detail for this. So if you are interested, you can search for how the eye muscle looks like. So how is the structure of oculomotor muscles. And there are also research concerning the whole body movement, which practically involve all muscles, skeletal muscles of the body. And this is definitely much more complicated. Okay, so I think we have done an overview of some basic anatomy and these connections about these neural loops. Now we are coming to the most important section of this lecture. So this is the second aspect I've talking about the mechanistic level. So how the human system or how the muscle is controlled. And we will also have an exercise later on this section. And this may also appear in exams. So this is important. So let's start with a physical mass spring damping system. So this structure is a typical basic structure which is usually taught in classical mechanics. I believe in the last lecture we also learned about this mass spring damping system. So they have a mass and they have elastic component, which is a spring, and a viscous component, a damper. The elastic component, the spring, generate force, resistant force to act against the displacement. And for the spring, we usually need to define zero length or resting length of the spring. This zero length means when there's no external force acting on the spring has a natural length, and this is called the resting length. And the viscous component, the viscosity, the resistance depending on the velocity. So that means when the system is moving, the viscous component always drags the movement, always slows down the movement. In the following, I will neglect the viscous component just to make things simpler. So we'll just talk about elastic component in this model. So the question is, does our biological muscle joint system also behave like a spring, like an elastic spring? The answer is yes, because as we have just learned, the stretch reflex, once you have a deviation of your muscle length, if your muscle length is lengthened, then through the reflex loop, you activate the muscles to resist, to make the muscle active and shorter to resist this lengthening. So this is already kind of a spring-like behavior. And also have to mention that not only just the reflex, but also there are passive properties of the muscle. So even without reflex, the passive muscle also act like a spring. But this is, we have to note that muscle only apply force in one direction. So if you voluntarily contract a muscle, the muscle only pulls, but you cannot generate a command to like a muscle push. And in this sense, the muscle is also different, like a spring in the traditional sense, because spring acts on the both directions. So we will please take a note, because in lecture we will in the exercise we will have a task about this point. So just be aware of the muscle is different from like a normal spring. So, okay, so but we still want to model the muscle like a spring. So at the spring, we have, let's make a plot of the spring. So we have length representing the length of the spring and the force of the spring. So this lambda, let's consider this as the zero length or the resting length of the spring. So this is actually a model of the muscle. So for the muscle, if you think it is a spring have a zero length, and if the length increase, and it exceeds the zero length, it will generate force to balance this increase of length. So here, for example, if your length is at L1, but your zero length is at lambda, and then the system will arrive at a point of force L. So this point is called the equilibrium point. It consists of both force and length component. And this is always in the static case when all the forces are balanced. It's not about dynamics, not about moving. So which means this force and length will define an equilibrium point. And once you are at equilibrium point, and if you are deviate anywhere outside this equilibrium point, the system will take you back to the equilibrium point. And that is the characteristic of the human motor system. So for example, the stretch reflex and many other reflex mechanisms. So it's kind of stabilized within a posture. And also we notice that if the muscle length is smaller than the resting length, there will be no force. The force will be zero. And in static case, this force, since the muscle balance the external force, so this force represents both external force and muscle force. Okay, and then you may ask why this characteristic has to be this kind of exponential curve. Like for example, the slope is very, very small at a smaller length change. When the length change is larger, the slope becomes much larger. So why it has to be like this, or not opposite, like this shape, or even linear shape? And this question we will deal with this in our exercise. But what I can tell you that the muscle has this kind of length, force length characteristic. It is based on the experimental measurement, because we can really measure this characteristic. And I will show you later. After explaining this modeling, I will show you some experiment showing how this curve is measured in the real human experiment. So from this figure, you need to know this lambda representing zero length and what equilibrium point is. And most importantly, this nonlinear curve of the muscle force length characteristics. So how do we produce a movement? Or how do we change the length of a muscle? So we can simply, if we want to shorten the muscle, we can simply decrease the zero length of the muscle. This curve, this exponential curve actually do not change much in shape. So let's assume they are the same shape, independent of where your zero length is. And actually, this is also confirmed by experiment. So when you shift the lambda to a smaller value, this force length curve also shift. So at the initial position, before the shift, the system was balanced at the length L1 and the load L. So this is the static case. However, once you change your zero length, and assuming external condition does not change, and then the same load will define a new equilibrium position L2. So after the shift, the system should have a new equilibrium, this EP2 here. And the previous equilibrium becomes a deviation from the current equilibrium point. And according to the previous figure, any deviation will be compensated by this reflex mechanism. So the system will automatically drag your position, your equilibrium point, to the current new equilibrium point here. And here we see that actually the shift, the movement is actually done as the emergent of shifting the resting length. So again, let me repeat again. So you first shift the resting length of the muscle, and this will induce a shift of equilibrium point. And then the previous equilibrium point becomes deviated from the current equilibrium point, and then the reflex will act to bring your previous your previous state to the current equilibrium point. And this will result in a length change of the muscle. Okay, I hope this is clear for you. And there's another condition about the passive movement. So unlike the previous case, the active movement, in this case, you are not changing the resting length of the muscle or the spring. So, but the external condition changes. For example, if you're holding a book in the hand, and then someone suddenly removes the book out of your hand. So what happens? In this case, the load decreases. And previously your system is stabilized at the equilibrium point. And because your load decreased to L2 and everything should arrive on this curve, the force length relationship, the equilibrium point should arrive on this curve. So this L2 will define a new length, a new muscle length here. So that means you will shift it from the previous equilibrium point to the new defined equilibrium point. So this explains a passive movement. So again, in passive movement, you are not changing resting length, but external load changes, and this leads to a shift of equilibrium point. Okay, so now we have been talking about a single muscle. As you see, this is length. Now, if we have two muscles, we need to consider a joint. So we need to have the joint angle and joint torque. So here we have a model, a very simple model. We have one joint and two opposing muscles. So F is representing for flexor and E is extensor. So here we have this force length relationship plotted in the torque angle space. So now this lambda is in the angular domain. And we have this characteristics of the flexor muscle. And the extensor muscle will have the opposite sign of the torque. So that's why it's drawing below the horizontal axis. And so here we see this lambda F1 and lambda Fe, the zero lengths for flexor and extensor are not, they are separate. So actually it can be somewhere else, but here they show there's some distance between the two resting lengths. But for this joint, if we want to move the joint to a new position, we need to move together the two resting lengths. So we shift the lambda simultaneously for flexor and extensor to a new position. So basically, it shifts the whole characteristic from the initial state to the end state. Here, I think this plot is some kind of confusing. Actually, it doesn't have to be connected. It looks one curve, but actually, they are two separate curves. So again, in this plot, we show that how you move a joint. You simultaneously shift the resting length for both flexor and extensors. And usually this is called a reciprocal command R, but that is not important. You don't have to know this terminology. So here is a shift of the lambda flexor and lambda extensor to the same direction to produce a movement. We also have a condition that the shift of resting length of flexor and extensor in the opposite direction. So here, I think I forgot to mention that you can have So this curve, this thick line is the sum of the two characteristics. That's why it represents the joint torque. And here, if we plot the sum of the two torque, flexor torque and extensor torque, we see when the distance between the two resting lengths, when the distance is increased, so the resting length is shifted in the opposite direction, we see this sum will have a larger slope. And this means that if we take at this point and take this slope at this point, it represents the stiffness of the angle at this equilibrium point. And once you have this shift in the opposite direction, you have a larger slope, which means you will have a larger stiffness. So again, to summarize, shifting the threshold of the two muscles to the opposite side, opposite direction, will increase your joint stiffness. Okay, and I know this is a little bit maybe not very easy, but you could think about it afterwards. And if you have questions on that, I can explain after this lecture. So from now on, it is less important. This is basically about the human experiment. And here you can see these different curves, these exponential curves is observed in different species and different muscles. So I can shortly explain how this experiment is done to get this curve. So the first done experiment in cat, and this is a very complex experiment. So they have to remove a certain area of the cat's brain to make sure that the cats do not produce voluntary movement. So and then they will stimulate somewhere in the brain to mimic the input, descending input to the spinal cord. And they take a muscle, like this is soleus, a leg muscle of the cat. They take a muscle and they when they give a stimulus, the muscle will be at a certain length. Actually, this length is somewhere in the x-axis, for example, here. So when you give a stimulus, the muscle will at this length, and then the experimenter will slowly stretch the muscle. So the muscle will become lengthened, and they simultaneously measure the force in the muscle. And in this way, when they stretch the muscle at different lengths, they can obtain different levels of force. So they make this plot. They can plot these individual points, and then they do interpolation and to show this exponential curve. And then they change the descending stimulus. So for example, they can increase or decrease. And this once they change the stimulus, the intensity of the stimulus, they will observe the resting length will change. So assuming that they arrive at another position. And at this new zero length, they will do the same experiment, stretch the muscle, measure the length change and the force, and then can still plot an exponential curve at this zero length. And we see for different muscles, with this shape is preserved across different resting lengths. Also in humans, it's we can do a similar experiment, but we cannot like do operations in humans. In humans, we ask the participant to hold a load in the hand, and then we will decrease the load at different level, and we ask the participants do not react to the change of this load. So we assume that the participants do not change the control signals to the muscles. So at each point, so at each position, for example, at this point, so we have this load, and then the experimental decrease the load to different levels, and we see the arm will arrive at different final positions. And until you completely remove the load, the arm will arrive somewhere along the x-axis, and that is where the resting length is. And you can ask the subjects to combine different loads and position, and you can plot a different curve. But again, this curve looks similar at different zero lengths, and you can also do this for extensor muscles. So, and for human eye muscles, you can also do this kind of experiment. And in this experiment, they implant a force sensor in the eye muscles of a patient, because a patient is undergoing operation, and they put a sensor to measure the muscle force. And then they ask the patient to look at different angles, and which is plot this x-axis as the eye position, and they simultaneously measure the muscle tension and also by passively stretch the eye muscles, and they also get this kind of exponential curves. So we see that this exponential nonlinear spring property is universal in human and cat muscles. This is a simulation study taking account to this force length characteristics. This is the scheme for one muscle control. So here you see this force length relationship. This is part of the model. The central command will be the resting length. But there are also more stuff like the gradual force generation of the muscle and also the passive muscle force generation depending on the passive stiffness and muscle velocity. And also they take into account the reflex delay in the model. And this passive property of muscle is actually more complex. And in this study, they use a simplified model. Actually, the most popular model we use a heel type model, which I will not talk it now, but Rashid will talk about a heel type muscle model in his lecture in 2 weeks. Okay, so this is just give you a general idea that we can do a simulation study using the model taking into account the force length properties of muscle force generation. So let me first briefly report what we are doing currently in our lab. So we have this model that I just introduced. So we have these equations based on the model that I present in the previous slide. And but in this model, we use the relatively simple reflex loop. So we only take the length-dependent feedback in the model. And here we have the two joint arm, which is a shoulder and elbow. And we have 6 muscles, 2 muscles only acting on elbow joint and 2 muscles only acting on shoulder joint, and another 2 muscles acting on both joints. This is called biarticular muscle, and for the muscle acting on the single joint is called monoarticular muscle. So we have 2 joints, 6 muscles. This is a biomechanical model, and this is the muscle control model. And we have this experimental recording. So in our movement lab, we have equipment to, we put the markers, reflexive markers on the joints. So in this case, we can measure the movement kinematics. And we have a display. We let the subjects do the reaching movement with the 2 joints from between different targets. And we also have a electromyographic recording system, which we put electrodes on the surface of the screen to measure muscle activations. And Rashid Ramadan has been developing an algorithm based on the optimization to estimate the control command based on this theoretical model. So we measure the output and we have a model, and we develop algorithm to estimate what is the input to the model. And while Cora Hummert from our group, she's also aimed to estimate the input of the model, but she used an analytical approach to mathematically invert this model and to calculate what is the input is. So it's mathematically very complicated. And I did another study. Still, also we are interested in what is the input to this system. And I measured the electrographic, the muscle activity. And I used a simpler model because when we come to the EMG level, so we don't need to take account into the mechanics. So I used the EMG to estimate, based on this simplified model, to estimate what is the input to this model is. So this is basically currently what has been done and what is going on in our lab. So in general, we are looking for method to explore what is the descending commands or the input control command to the model should like. And here I would offer a master's thesis. This is related with a research program that I'm currently working on. So if you are interested, please contact me through email, and I would be happy to talk with you and talk about the details. Let me explain shortly what the task will be for this project. So as I said, we use a relatively simplified reflex loop in our model. So now we are trying to integrate a more complex reflex to the model. For example, the reciprocal inhibition as our first step to add to this model. And of course, later on, we will add more reflex loops to this model. And the second task is we are developing algorithms to estimate descending command. So as Rashid did, they use optimization method, but we are still working out if there's solutions. So given a mathematical model and we can measure some behavioral data, how can we estimate the input to this model? So these first two tasks is more important for this program. But there are another two options that could be done depending on the interest, on the applicant's interest. So we could do design some behavior experiment, because we have here you see we have these markers we can put on the human limb, and when the human doing movement, we can measure the movement and make a plot to see how movement look like. So this is kind of more like experimental study, not behavior recording. So this is one option. And the other option is there's a software called OpenSim. This is very advanced simulation environment that they take a lot of biomechanical details of the musculoskeletal structure. So they have very precise parameters of bones and muscles. So they can even completely simulate all the muscles, in this environment. Because previously we have done everything in MATLAB, so now we are thinking to use this software, which may be more convenient. So this is one option for us to explore. Okay, so this is about the research program. Again, if you are interested, please contact me and we could discuss. So, okay, I think we are close to the end. Still, okay, earlier than I had expected. So let me shortly summarize what I have learned today. So at the beginning, we have learned about the muscle structures, and we see how the muscle generate force through these muscle fibers and these action potentials. So, and then we look at the sensory part, the muscle spindles, which is give you a muscle length feedback. And then we went through several reflex loops. And there are many types of reflex, and these reflex modulate the motor output. And the last, but most importantly, the mechanistic level, we model the muscle as a nonlinear mass spring model. So we have to understand how this model works to generate different types of movement. Okay, I think we are done for this lecture, and for next lecture, I will talk about even higher level about the brain, which is more complex. So, okay, maybe I will stop sharing the screen.